Okay, now we're getting to the good stuff. Cellular respiration. I don't know if you're as excited as I am, but hey, um, the only thing about cellular respiration, um, this is going to require some memory, okay? And we'll get into that in a little bit. So let's move to the next slide. All right, so carbohydrates, fats, and proteins, we know that all of these can be used as energy sources, okay, for us. And we know when we're talking about a metabolic pool, we're saying that when we take in these food sources, we're saying when these food sources are broken down, because if you remember from chapter 1, we talked about catabolism. Catabolism meant that we were taking something bigger and breaking it down into smaller um, parts. So that's what I mean by catabolism. So, and what this says is that catabolism produces molecules that can also be used for anabolism. So it says some of those products that are broken down can be used to build other compounds, okay, or bigger things. So catabolism, we know, is the breakdown of molecules into smaller usable parts. We know that anabolism is when we synthesize or build, take smaller things and build them into bigger things. So it says when these carbohydrates are broken down, when these fats are broken down, when these proteins are broken down, it says that they can be used for energy sources. Why? Because when we break bonds, if you remember, we release energy. All right, so cellular respiration is then viewed as this complex series of catabolic reactions in which the energy of food molecules, whether they're carbohydrates, fats, or proteins, it says this energy can be transferred to ATP or transferred to building ATP. And we know that ATP is built from adenosine diphosphate or ADP plus inorganic phosphate. Okay, so it says when we break down um, uh, glucose, for example, because that's the example that we're going to use, but it says when we break this down, it says when these hydrogen ions and these electrons are being lost, it says we can use that energy eventually to build ATP. So when we're pumping or allowing those hydrogen ions, well, actually, we're going to pump them across a membrane during the electron transport system. Specifically, the membrane that we're going to be pumping them across is called the cristae of the mitochondria. And when they are able to flow back across the membrane, then that's going to give us enough energy to take ADP plus P in order to create ATP. But we'll get into that later. But we just want to know catabolic reactions are reactions that release energy. Okay. All right. So here's the structure of the mitochondria. If you remember, the mitochondria is a double membrane structure. Usually the outermost membrane is the membrane that where it was captured in a vacuole and put to work for eukaryotic cell structures. The inner membrane we see is folded or convoluted into what we call the cristae. And then the other thing that you'll see is that there's actually a space between that inner folded membrane called the cristae and the outer membrane, and that space is referred to as the inner membrane space. We're going to find that's very important because when these hydrogen ions are being pumped across the membrane, they're going to move into that inner membrane space. Then if you look at the inner area between the convoluted folds of the cristae, you're going to find in this picture is actually colored purple. That would be the matrix, and that in the matrix is where those coenzymes, the nicotinamide adenine dinucleotide and the flavine adenine dinucleotide are going to be uh, this is where they're going to actually drop off those hydrogen ions and electrons so that they can be used to during the electron transport system. But we'll see and talk about that a little bit later. So the areas that are mentioned here, it says the cytoplasm. This is where glycolysis. So we're going to find out that cellular respiration is broken down into reaction groups. And so we begin with glycolysis. So it says, where's glycolysis going to take place? In the cytoplasm. And then we're going to move to the transition reaction, or sometimes it's referred to as the preparation reaction. So here, this is where it's going to take place in that purple area called the matrix. Also taking place next would be the citric acid cycle taking place in this purple area. And we'll find out that the citric acid cycle actually has another name. And then that convoluted membrane is where the electron transport system is going to be located. And that membrane is called the cristae. All right, so where were mitochondria proposed to have come from? Same thing as chloroplasts. So what I want to talk about are some of the reasons why they say that mitochondria came from prokaryotic organisms or bacteria. All right, so what's some of the evidence? It says if you examine a mitochondria or even a chloroplast, they actually have their own circular loops of DNA, which is the same thing that prokaryotic cell structures have. They also have the 70S ribosomes. Eukaryotic cell structures have 80S ribosomes. So it says that these guys also have the 70S ribosomes. And one of the other things is that because they have their own DNA, it says that they can reproduce independently of the cell's own reproduction. So this is other evidence saying that they arose from a prokaryotic cell structure. They are also killed by some bacterial antibiotics. So again, this is alluding to the fact that they arose from these prokaryotic cell structures, for example, the bacteria. And then it says, evidence suggests then that they are symbiotic bacteria cells. And we know that the word symbiotic means living together. So in other words, we know that some bacteria can harm us, but it says that these guys are symbiotic. So they're not going to harm us, but they're actually, the two of us are going to actually coexist together. Okay. So that's what that word symbiotic means. So it was proposed that these bacteria cells were captured in a vacuole and put to work making ATP for eukaryotic cells. All right, so let's look at the equation. And the only thing that I can't see, and maybe it will show up eventually, oh, there it is the arrow. All right, so, and we know that the arrow always means yields, okay? So it says if we take glucose, which is C6H12O6, plus 6 oxygen molecules, it will yield or produce 6 carbon dioxide molecules plus 6 water molecules plus ATP. Okay, and the only thing that this reaction doesn't say is if we broke down glucose, it says it doesn't tell us the exact amount, which is 36 to 38 molecules of ATP. So one of the things that we saw at the beginning of that reaction, and that was glucose and oxygen. So those were our two reactants, or those were our substrates, where it says that oxygen is going to fuel cellular respiration. So it says a steady stream of oxygen passing across that membrane is going to fuel cellular respiration. So where do we need the oxygen? We actually need oxygen during the electron transport system, because oxygen's job will be to pick up those energy spent electrons from the electron transport system so that we can fully and completely break down glucose. All right, so we've already mentioned the reaction groups, but let's go over those reaction groups again. All right, so the first of the reaction groups is glycolysis. We said earlier that it occurs outside of the mitochondria in the cytoplasm of the cell. Okay, so this is outside of the outer membrane of the mitochondria, which would make it occurring in the cytoplasm of the whole cell itself. Then we move over to the transition reaction. The transition reaction is also called the preparation reaction. It occurs in the matrix of the mitochondria. So remember, that was that purple area that we saw. Not that it's purple, but that was just so that you could see the area. And then we move over to the Krebs cycle, or the citric acid cycle. That also occurs in the matrix of the mitochondria. And then finally, the electron transport system. The electron transport system occurs on the cristae. One of the things that I would like you to remember also as you move through these different reaction groups, and that is the fact that there's some things that you need to remember. You need to always remember, what is the beginning substrate? What did we start with? Then you want to know, what is the ending substrate? What did we end with? Other questions that I would ask is, did I use any energy during the particular reaction group? Did I gain any energy in the particular action group? And then the other thing that I would look at is, which of the nucleotide coenzymes did we use? Which coenzymes did we use? Did we use NAD? Or did we use FAD? Okay, so which of those two did we use in order to pick up the electrons and the hydrogen ions that are being lost during the breakdown of glucose? Okay, so again, those. Right, so glycolysis. Now, we know that glycolysis, it translates as a sugar splitting reaction. Glyco referring to it being sweet, lysis meaning to split. These are words that you learned before. So glyco again, referring to the sugar itself, or sweet, and then lysis meaning to split. So what's happening there? So this is just a preview before we actually get into all of the guts of glycolysis. And again, I'm giving you the highlights and some of the important things that you need to know, but not every single thing that's taking place in this particular chapter. Okay, so it says glucose is split into two 3-carbon molecules, and so at the end of glycolysis, we're going to find that we have two pyruvate molecules. Those are the two 3-carbon. So we know that this is a sugar splitting reaction, because at the end, even though we started with one big molecule called glucose, it says at the end of glycolysis, we're going to have two 3-carbon molecules. Again, this guy's believed to be one of the oldest group of energy releasing reactions, and the reaction is taking place outside of the mitochondria, and not inside of the mitochondria. And so again, it's outside, occurring in the cytoplasm, not in a separate organelle. Aerobic means that this reaction can take place in the presence of oxygen, and it could also take place in the absence of oxygen. So the word anaerobic means in the absence of oxygen, and the word aerobic means in the presence of oxygen. So I will see you in the next video. Have a great day.